Good afternoon and good morning wherever you are, and welcome to the first seminar and webinar and scientific discussions of the Genome India International. We have an excellent speaker today to start with our lecture series. Before going to the starting the lecture and introducing Christine to all of you, we welcome you all participants again, and we will first request the founder and current president of Genome India International, Professor Chitranjan Kole, to give his remarks and comments, and then after that we will start the lecture from Christine. Professor Kole, please.
Thank you, Dr. Pandey. As President of Genome India International, myself welcome the speaker today, Professor Christine Helen Foyer, and our today moderator, Dr. Monish Pandey, and all the audience, and definitely members of Genome India International. The members of Genome India International were thinking to start one webinar series with dignified speakers selected from all over the world. And we are very happy today to have you, Professor Foyer, as our first speaker. And we expect it would be very exciting and enriching talk. So without wasting much time, I would request Dr. Pandey to proceed to introduce you and start today's webinar.
Thank you, Professor Kole, for your nice words. So thank you. And Christine and others, if you are not aware, so basically Professor Kole is the one in my PG studies who drive me to science. So basically you can say he diverted my road, and then I took that road and where I am today now. So now to give one or two sentences like information about GII, already Professor Kole mentioned, but I would like to reiterate that it is established in 2005 and has been serving as platform for networking, knowledge sharing, and building partnerships by engaging researchers, academicians, private entrepreneurs, media, and national and international funding agencies involved directly or indirectly in genome science-based solutions. Now, we are starting actually GII lecture series, and really we are honored to have Professor Christine here. Really, and we are very much thankful that she has agreed to be our first speaker. She is internationally renowned researcher, academician, and policymaker to start this lecture series. And today's presentation title will be Roles for Reactive Oxygen Species in Plant Growth and Defense. Although you can find, if you just type name of Professor Christine Foyer, you will get a lot of information, but we don't have that much time to say good words about her. So you please search in Google about her and her accomplishments in detail. I could only say summary of her that she is currently professor of plant sciences at the University of Birmingham, UK. She is the president of the Association of Applied Biologists and editor-in-chief of Food and Energy Security. She currently holds the Janaki Ammal Chair of the Indian Academy of Sciences and is a member of the French Academy of Agriculture. She is also a senior editor for Plant, Cell and Environment Journal, and is also an associate editor for many journals, and few, if you want to name, Biochemical Journal, Journal of Experimental Biology, and Physiological Plantarum. Professor Christine Foyer has published, more than 450 publications. I know you she made a century of centuries. So you can say 100, she has H index of 102. So you can say 102 papers are cited 100 times. So 100 centuries. And we congratulate her also for all these accomplishments. She has currently more than 82,000 citations. She has been listed, of course, in most of the science-based listings where they list top researchers, highly cited researchers in the world, and she is one of the science leader in our international research arena. If you want to see what is her expertise, so she is an expert in plant metabolism and regulation under optimal and stress conditions. Her lab focuses on the role of reduction oxidation processes, signal regulate plant growth and stress tolerance, studying how primary processes under the redox status of cells and associated phytohormone signaling under optimal and stress conditions. So, and she is using model Arabidopsis as a model, and other crop plants also she is working like wheat, barley, maize, soybean, and tomato. And this is like a research interest that is pursuing her lab, but I know that she has been engaged in many of the consortium initiatives and policy making agencies to drive the science to the next level. So I would request, let's welcome Professor Christine again and listen her talk on this interesting topic. Please feel free to drop your question in the text box, and we will try to accommodate at the end of the presentation. So Professor Christine, we welcome, and the floor is yours and for your presentation.
Thank you. I will now share my screen. I hope you can see it.
Yes.
Yeah.
Okay. So it's a great pleasure for me to talk to you today. I always enjoy talking about reactive oxygen species. I started when my PhD working on the role of antioxidants in plants when we knew very little about the topic. And it's amazing to say all these years later, this is still an area that is growing in interest. So over the intervening years, we've learned a great deal of knowledge about what happens in terms of the production and use of reactive oxygen species in signaling, and our knowledge continues to grow. So I'm going to give you a very basic introduction for those who are perhaps not so familiar. I will talk about some of the work that we have been doing in the lab, and I will then finish on how we understand the role of reactive oxygen in cell-to-cell signaling during stress, because I think this is particularly important for anybody who is working in the area of acclimation to a changing environment. So I will start at the very beginning. So this is just a stylized cell, and this simple diagram is just to illustrate that all of the compartments of the plant cell produce reactive oxygen species. Reactive oxygen species are part of the normal metabolism of plants. So we have production within the chloroplasts as a result of the photosynthetic electron transport chain, producing superoxide and then hydrogen peroxide. We have the peroxisomes, which produce enormous amounts of hydrogen peroxide, particularly as a result of photorespiration in green tissues. And then we have mitochondria, again, which are producing superoxide and hydrogen peroxide as a result of respiratory electron transport. And there are other reactions in the cytosol, etc., which together produce an enormous amount of metabolic hydrogen peroxide. In addition, and most importantly, we have enzymes on the plasma membrane and also in the apoplast cell wall environment, whose job it is to produce superoxide and hydrogen peroxide. Of particular note are the NADPH oxidases, the respiratory burst oxidase homologs, we call them. These NADPH oxidases function to produce a burst of hydrogen peroxide in the apoplastic environment in response to any physical or chemical shock. So here we have these enzymes who simply produce hydrogen peroxide in response to any kind of chemical or physical stimulus. So that is the environment where we have huge amounts of production of hydrogen peroxide, both in the cellular environment as a result of metabolism and in the apoplastic environment as a result of cell signaling. We used to think that a lot of the production of hydrogen peroxide within organelles such as chloroplasts was uncontrolled, that it was just by virtue of the fact that when you have electron transport chains in oxygen, you have escape of electrons to produce superoxide and hydrogen peroxide. But in recent years, we know, for example, here is the photosynthetic electron transport chain, which, as you see, has many points where you can produce superoxide and hydrogen peroxide. We now know that actually the production of reactive oxygen here is tightly regulated, and each of these complexes is able to function in a regulatory sense to limit or allow the production of reactive oxygen. So it's a much more controlled metabolic production of hydrogen peroxide than was at first envisioned in the past. And of course, once you have the production of hydrogen peroxide, we have to have systems which remove or process hydrogen peroxide and other reactive oxygen species. And here we have two models of how this happens. The first model is very simplistic, and it has served us well over many years. The idea that you have a good balance between reactive oxygen species and antioxidants here in red, that is maintained in what we would call cellular redox homeostasis. However, in response to any stimulus or stress, reactive oxygen species are produced in greater abundance, and so we have an imbalance between antioxidants and oxidants, which leads to either stress or signaling. So this is kind of the very simplistic approach where we would think of here going to oxidative stress or oxidative signaling. This was based on the idea perhaps that low amounts of reactive oxygen species are involved in signaling and large amounts are not. They are simply involved unwanted, and they're involved in perhaps negative processes. However, this isn't a correct hypothesis, because any reactive oxygen species can act as a signal. There is no distinction between different reactive oxygen species. Whether a reactive oxygen species acts as a signaling molecule or is removed by an antioxidant depends on where the reactive oxygen is produced and the availability of other processing molecules within that environment within the cell. So here in model 2, we have a very different idea of how reactive oxygen is processed, where you can have many pathways of processing or removal, depending on how the reactive oxygen is produced, where it is produced, and whether there is a processing molecule, such as a peroxidoxin or a thyrodoxin close to it, or an antioxidant. And so you have multiple pathways and multiple fates. So you can have acclimation or cell death, depending on how the reactive oxygen is perceived and processed in any compartment of the cell. So it is a very much expanding idea where you now have a range of processing and sensing molecules for hydrogen peroxide. So on that basis, we are starting to understand how signaling from hydrogen peroxide works within cells. So as I told you, we have production of reactive oxygen in different cellular compartments, but in each of those compartments, there are also proteins which act as receptors for hydrogen peroxide. We didn't know much about these receptors until recent years. And the first of those receptors that was described is called HPCA1. HPCA1 is a protein that resides in the plasma membrane. It is a protein kinase, and it is able to perceive hydrogen peroxide and change the redox signal into a phosphorylation signal within the cell. So the perception of hydrogen peroxide in the apoplast is then transmitted into the cell to change gene expression through a phosphorylation signal. And this was the first to be described, but now there are other redox sensors and an accumulating amount. I've just put a reference at the bottom to another redox sensor, which is inside the cell. This is QS-SOX1, which is again involved with transmission of signals. And there are other types of proteins now in different cellular compartments, which are described as redox sensors that sense hydrogen peroxide and can transmit information in a very specific way to the nucleus. So we have an evolving situation of understanding of how reactive oxygen species are signaled as well as removed by antioxidants. And now you see that you have reactive oxygen involved in signaling, and any excess reactive oxygen is removed by antioxidants to limit the spread of the signal. We use a lot of systems to detect reactive oxygen species, and it has been a real problem over the years because it's not easy to accurately determine the level of reactive oxygen. Often people use methods which measure the level of reactive oxygen across the whole cell. That tells you nothing about compartmentation. And as I mentioned to you, a lot of reactive oxygen is actually produced in the apoplast cell wall compartment. And when people measure reactive oxygen, they often assume that the amount they've measured is actually within the metabolic space, when in fact possibly it isn't. So it is difficult to accurately get an idea of the level of reactive oxygen species in individual cellular compartments. But one of the best ways, and it's a methodology that is continuously improving, is to use in vivo genetic probes. And this is one of the most simplest that has been used for a number of years. And this is called redox sensitive green fluorescent protein. So green fluorescent protein is well known as a marker molecule. And within RoGFP, there are cysteines which have been engineered into the proteins, which are redox sensitive, and they can undergo a glutathione reaction in response to hydrogen peroxide, providing a quantitative measure of the level of oxidation at any moment in time. This green fluorescent protein can be targeted to most compartments of the cell. The only compartment that is not possible at the moment is the apoplast cell wall. But using these probes, we get a much more accurate idea of the changes in oxidation state which occur in response to stresses or different environmental conditions. And I will just show you some data. So here we're looking at cells, Arabidopsis epidermal cells, expressing RoGFP. And this RoGFP is targeted to the cytosol, but it also goes into the nucleus. So you can see in the epidermal cell, and we measure two wavelengths which together allow us to measure the oxidized and the reduced state. So the oxidized and reduced state, and we measure a ratio of those parameters, which gives us a quantitative measure of the oxidation state. So this can be targeted to different cells, and you see here stomatal guard cells in the epidermis. You see again the green fluorescent protein in the cytosol and the nuclei very, very clearly. So with that information, you can apply different stresses and find out what's happening. Now, this is an experiment that I did with colleagues from India. I had a student, Risha Babu, who came from Anil Grover's lab to work in my lab, and she was interested in heat stress. And in this simple experiment, taking Arabidopsis seedlings, which are expressing Rho-GFP, they were exposed to a heat stress of 42 degrees centigrade for 60 minutes. After that, we measured the redox state of the cytosol and nuclei in the epidermal cells and the stomatal guard cells. And if you just look, you see we measure the ratio at the top, and that's not very useful to you. But from that calculation, we can measure the degree of oxidation. And when we looked at these data, you see the white bars are the absence of stress, and the black bars are after the imposition of stress. You see there is an extensive oxidation of the cytosol, but surprisingly, a very strong oxidation of the nuclei. Now, this was the first time that this was demonstrated. Oxidation of the nucleus is surprising because there are no real known reactions in nuclei which will cause oxidation. So this demonstration is very interesting. Since we did these experiments, colleagues have repeated them. So it's clear that oxidation of the nuclei, particularly in heat stress, is a real phenomenon. And actually, oxidation of the nuclei occurs before oxidation of the cytosol. Now, when you have this kind of knowledge, it changes the way you think about the way stress works, because we often think of stress in the chloroplast, the cytosol, we're sending signals into the nucleus. If you have direct oxidation of the nuclei, you have direct regulation of transcription factors in the nuclei by redox processes, which can be very, very rapid, as well as all of the epigenetic factors which are present in the nucleus. So it gives a new level of understanding, which we are now looking at in greater detail to understand how this functionality works. But just to take these experiments a little bit further, you probably know that the response to heat stress is very, very specific in terms of gene expression. And the antioxidant system is regulated only in a very specific manner. So we know that oxidation, as I've showed you, is a major effect of heat stress, and that oxidation triggers heat shock factors, etc. But there are very few antioxidants that are expressed in response to heat stress. This slide just illustrates how specific it is. So these are the ascorbate peroxidases, different ascorbate peroxidase genes in Arabidopsis. The only one which is increased in expression is APX2. And that is really surprising. There are very few other changes in enzymes such as catalase or superoxide dismutase. There's a very, very specific activation of ascorbate peroxidase 2, which suggests that it has a very specific role. In addition to that, we took the studies one step further, and I'm not going to show you the data, but please look at the paper, because not only is there a change in the expression of APX2, we also have a post-translational modification in response to heat stress. And that is regulated by a calcium-dependent protein kinase called CYP28, which we showed in this paper directly interacts with APX2. to phosphorylate it. When we looked at mutants in both APX2 and CYP28, we found they were more sensitive to heat stress. And when we looked at the interaction between CYP28 and APX2, we could see direct phosphorylation on two specific residues on the protein. So both the response to heat stress in terms of oxidation and the response in terms of antioxidants is very, very specific. And while we know a lot about heat stress, I hope these data have showed you there are a lot more that we actually need to look at in terms of the intricacy of regulation. I just want to move on because I've just shown you leaf tissue, but of course you can measure all of the other tissues using this technology. And it's the effect of stresses is particularly important in flowers. One thinks of the susceptibility of flowers to heat stress and the reproductive tissues. So we can here show that we can measure the degree of oxidation in all of the tissues of the flower using RoGFP. And when we use this technology, we can look at how oxidized each of the tissues are under different conditions. In these experiments, we looked at the oxidation state of the flowers, just in the wild type plants, but also in plants which are deficient in glutathione. So the wild type is in black, and plants deficient in glutathione, which is an important antioxidant in plants. You'll see data here for the various tissues of the flower. Most of the tissues in the wild type plants are reduced. You can see that here. This is mature pollen, this is germinating pollen, and this is the pollen tube. What was a surprise to us really was that for all the tissues in the flower, the mature pollen is highly oxidized in contrast to the other tissues. So you have a highly oxidized mature pollen that has to be reduced in order to germinate. So as like many quiescent tissues, there is a level of oxidation in the mature pollen, and reduction is essential for activation, germination. When you take away an antioxidant like glutathione, all of the tissues become more oxidized. But you can see here how very oxidized the mature pollen becomes, more than it is even in the absence in the wild type plants. It becomes reduced following germination, as you see in the germinated pollen here. But there is a loss of function here, and many fewer pollen germinate in the absence of glutathione. And you see here, even in the germinated pollen, they remain relatively oxidized. So now we are taking this forward and actually looking at the effect of heat stress on these various tissues and processes to see how far this affects how the tissues germinate. I want to move on now to another set of experiments that I think you might find interesting. And this involves plant growth promoting microorganisms. We started this work in my lab because a colleague in Spain had isolated a group of bacteria, Pseudomonas habitans, from trees, from fruit trees, in fact, from plum and peach trees. And they found that these bacteria were very important in promoting the growth of the roots of these trees. And we know from the literature now, there's a growing body of evidence of the importance of plant growth promoting bacteria. There are rhizobacteria, but there are also fungi which are involved in promotion of growth, and we also have mycorrhiza. And so the soil microbiome is rich in organisms which can contribute to the growth of plants, and we are starting to learn more about how they work. We don't know a lot though about how reactive oxygen species are involved in these interactions between organisms. The action of growth promoting bacteria can occur in many ways. So we know there is a basic effect on the nutrition of plants by improving the uptake of nutrients such as nitrogen or phosphorus. Organisms in the soil can actually protect against infection from other organisms by acting antagonistically to them. They also produce, very importantly, plant growth promoting hormones, which affect the growth of the roots. And they can certainly, by interacting with roots, engage with the plant's defense systems to improve defense against other organisms. So we have a range of important interactions. So in the studies that we did and we reported recently, we went to Arabidopsis as a simple system to understand how roots responded to this organism, Pseudomonas oryzahabitans. When we started the experiments, we actually put the bacteria around the roots. When you do that, though, it's very difficult to see the interactions because the bacteria grow prolifically on the roots and they almost strangle the roots because the root exudates are so much. So you get a really strong growth of bacteria that does very much inhibit the growth of the roots. So we took a different approach. We asked what signals pass between the bacteria and the plants when there is no contact, no direct contact between the organisms. So we grew the Arabidopsis plants for 6 days, and then we transferred them onto plates where the bacteria was placed at least 5 centimeters distant from the roots. And then we measured the root response 7 days later. And it was quite clear there is a very direct effect on the roots, even without a direct contact. So the sensing of the presence of bacteria changes the root growth. So there is a proliferation of lateral roots and a decrease in the primary root, so that there is a large increase in lateral root density. So the perception of the bacteria increases lateral root density very, very clearly. We wanted to know how this happens. So we took the approach of doing the transcriptome analysis of the roots. We only did it at one time point, which is the time point of 7 days when we could see the change in root system architecture. But we were interested to know what transcripts were changed. And here you see some of the data. And the surprising thing is that relatively few transcripts are changed. And maybe this is because there is no direct contact. So it has to be the transmission of signals across a distance, which changes gene expression in the roots. And we wanted to know what types of genes were changed. And the first thing to notice is there is very little response to stress. So at the top, you'll see response to stimulus, response to stress. There were a couple of genes changed, but very minor numbers, very little. So as you go through response to other cellular stress responses, there is something, but not a huge amount. The greatest response that we saw were changes in response to oxygen and changes in metabolism that occurred in response to perception of the bacteria, which was really surprising. So without any contact between the roots, you get this change in the way that the roots are taking up nutrients. So these are just the most expressed or the most decreased transcripts. So if we look at the ones which are expressed first here, there's interestingly a heat shock factor here, but most of these, for example, this gene here is a potassium channel, and there are genes such as this one, which is involved in nutrition. There are some ethylene response transcription factors, but there is nothing which is like a response to a pathogen or a response to severe stress in the genes that are highly expressed. The genes which are decreased in expression, interestingly, include a lot of UDP glucosyl transferases, which suggests that there is a change in metabolism which is going on in response to the perception of the roots. And I'll just show you a selection of transcripts. So we were very interested in the hormones that were changed. And surprisingly, we were looking for auxin, we were looking for jasmonate. We didn't really see any of that. What you can see is a lot of transcripts which are response to ethylene. And ethylene response transcription factors. There is only one auxin transcription factor here, which we saw, which was changed. The other group of transcripts are response to changing oxygen levels. And perhaps this is not surprising, and we don't often think about this, but when you put organisms together in the soil, they do compete for oxygen. It's normal, and people don't even think about that. But however, you get these oxygen responsive genes which are expressed, but they are not expressed in such a way that the total root mass is inhibited. But we do have a hypoxia response. And then we have this whole suite of genes which is involved in nutrition. So there are lots of transporters which are changed in expression, and there are lots of root hair specific genes which are changed in gene expression, as you'll see here. There's even a sucrose transporter here, a boron transporter, as you will see here, and other types of transporters which are important in the nutrition of the plants. So the perception of the bacteria is changing the nutrition of the plants without even touching them, which is really a very interesting observation. However, all of that data, while interesting, didn't tell us anything about the role of reactive oxygen species. It did tell us there was no great oxidation in response to the presence of bacteria, which was interesting. So we took a different approach to ask, how important are reactive oxygen species and redox signaling in the plant response to the bacteria? And the first approach we took was to look at the role of a transcription factor, which is called redox responsive transcription factor 1. It also has another name, ethylene response factor 109. It is a really important transcription factor because it's important in the perception of reactive oxygen species and the amplification of reactive oxygen species, as well as the sensing of jasmonate and other hormones to regulate root architecture. So we have different types of plants. We have plants overexpressing this transcription factor, and we have plants that are missing this transcription factor. And we again ask the question, how do the roots respond to changes in this transcription factor? You can see, if we look just at B here, you see the normal response of the roots to the bacteria, the gray being the presence of bacteria. This is lost when we overexpress the transcription factor or it's missing. In fact, in the mutant TRF109, there is even a tendency to go in the opposite direction with regard to root architecture. So definitely this transcription factor is important. We then asked, if we take away antioxidants, what happens? And first of all, we took plants which are missing ascorbic acid. This again is the wild type. You see the lateral root density in response to bacteria. We have 2 mutants here which lack ascorbate, and you see the response is still present. So this tells us there isn't a general oxidation which is important in this response. However, it is a specific response, because if we take away glutathione, then you see here the response in the wild type again, and 3 mutants that are deficient in glutathione the root response is lost in response to the absence of glutathione. So this tells us the importance of glutathione. We then asked, what is the signal? Is it a volatile signal, or is it a signal which is moving through the physical phase of the medium between the plants and the bacteria? So we had lots of controls, but we physically separated plants and bacteria in different ways. You see here, the plants are on one side, the bacteria are on the other, with a clear gap in between them. And we could see without a doubt that if we separated the plants and bacteria in different ways physically, then we lost the effect on lateral root density. So the signal is not volatile. The signal must pass physically from the agar to, through the medium, from the microorganisms to the plants. What we know about the role of redox processes in root architecture is kind of summarized here. And this is our redox responsive transcription factor 1, ERF109, which we clearly show is important in the response of lateral root growth to the bacteria. So this factor and its role in amplification of ROS is important. We also were able to show that glutathione, which is known to be important in the growth of lateral roots through various glutaredoxins, is also required for the response to the bacteria. So the bacteria are sensed and altering lateral root architecture through the systems which normally use redox processes, but there's no real extensive oxidation which causes the change. We still have quite a lot to do. This is kind of the common model that people have of how root architecture responds to environmental cues here. And often it's considered, for example, that jasmonate is involved, and the expression of jasmonate related factors leads to the expression of auxin biosynthesis genes. Here's our ERF109, which is critical for that response, leading to auxin signaling, which changes root system architecture. Well, we have our environmental cue, which is our bacteria, but we see none of the expression of jasmonate related factors. We know the importance of ERF, but we know that perhaps now there is a different route that doesn't involve the jasmonate so much in the changes. So we have to look more closely at how this process happens. I want to now just talk about general reactive oxygen signaling and how plants perceive reactive oxygen. So it's considered that in the responses of roots, the response to environmental change and the presence of organisms such as bacteria occurs at the root cap. The root cap is where the signal is perceived, and the signal is transmitted from there throughout the plant. And I want to just talk to you now about how ROS acts as signals in response to the perception of any kind of stress. In our study there, we were looking at the presence of bacteria. But these are slides that were kindly provided by my colleague Ron Mittler, which summarize a lot of the data that was obtained in his lab and my lab and many other labs. But it will give you an idea of the way that ROS signaling works. So, as you know, all plants within the environment are exposed continually to a range of biotic and abiotic stresses. They have to react quickly through a stress signaling mechanism, which transmits signals from the point where the stress is perceived throughout the whole plant, and also from plant to plant. this signaling is really, really important. We've known for a lot of years that signals are transmitted through the plant. So you can have local signaling, for example, at the point of perception of a light stress, and that signal here in Arabidopsis is rapidly transmitted to all the other parts of the plant. So now we know a lot more about this systemic signaling. There are different types of processes involved. Of course, you have to have hormones, and you have electrical signals, calcium signals, and waves of reactive oxygen species. And these are involved at the propagation of signals throughout the plant. And I want to really focus on Ross waves and its interaction with calcium waves here. So this is the way that Ross waves are transmitted. And this is a model. Sorry, is this all right? You can still hear me. So on the perception of stress, there are signals which pass from cell to cell as a wave of reactive oxygen species, like this. So the perception of a stimulus triggers a calcium burst, which activates the NADPH oxidase, the respiratory burst oxidase homologue here, on the plasma membrane. This produces a wave of reactive oxygen, which can then re-enter the cell and move from cell to cell, and it also moves through the apoplast to the next cell. But this transmission of signals moves from cell to cell to cell in a Ross wave. Now, the importance of this process has been demonstrated in every kind of abiotic and biotic stress. So these Ross waves, in my own lab, we've shown them to be really important in CO2 signaling, but any kind of light signaling, heat stress signaling, pathogen signaling, wounding signaling, has been shown to involve this type of Ross wave. And I'm just going to show you some examples. This is one of the nicest that I know. This is the closure of stomata in response to excess light. And in these experiments, look, excess light was given to a local leaf here, and then the closure of stomata on this leaf and every other leaf on the plant, you can see them labeled here, was measured. So you see the closure of stomata, and here is the control at the top for the local leaf. When you apply highlight to the local leaf, you see a rapid closure of stomata within minutes, which is more surprising is that if you measure the distal leaves, which have never seen highlight, the closure of stomata is nearly as rapid. And you see here the way on different leaves, the closure of stomata occurs. So there is rapid transmission of the signals to close the stomata. And this requires Ross waves. Now, the way that this has been done has been very elegantly demonstrated. This is an apparatus used in Ron Mittler's lab to image, but any imaging system can do it. We can do it ourselves under a confocal microscope with time. But these kinds of systems are used all the time in animals, and they're now being transferred to plants. So taking what we know about responses to stress and looking at transcription factors which are expressed in response to stress, we can take the promoter of the transcription factor and couple it to a green fluorescent protein to, for example, look at the spread of the signal. And this is a beautiful example of the spread of Ross wave. From here, the imposition of the stress, you see it here, and you see how ZAT12 is expressed quickly and rapidly throughout the plant. So you have this rapid transmission of signals moving throughout the plant to control gene expression in response to a local stress in tissues that have never seen the stress. Okay, now I just want to show you some of the data that shows very clearly that reactive oxygen species and the respiratory burst oxidases are essential for this type of signaling. So in experiment at the top here, we have plants which are exposed to highlight. We have the wild type controls and plants underneath that are exposed to highlight. You see here the control, and then you treat one leaf to highlight. You see this rapid transmission of signals. This is wounding the second one, control at the top. You wound one leaf. The transmission of signals via the expression of transcription factors is very rapid. And here is a pathogen. So different types of stress, the control, the pathogen. So this systemic signaling is vital to all forms of stress. And we know it's important because we can do the same experiments in mutants which are lacking components such as the NADPH oxidase. So here we have the imposition of stress, and here we have mutants lacking one of the forms of respiratory burst homologs, the NADPH oxidases. You see the rapid systemic response in the top and its near absence in the mutant. We can do the reverse. We can take away one of the antioxidants in the system. So here we have plants which are mutated in one of the ascorbic peroxidase forms, APX1. You see the transmission of the signal here, and its amplification, it's more rapid in the absence of antioxidants. So we can also do this just looking at the ROGFP signal. So we can measure oxidized and reduced ROGFP. This leaf is wounded here. And with time, you can see the spread of the oxidation, just putting the ROGFP system under a time-lapse photography. So based on this type of technology, we now know many factors which are involved in this propagation of signals. We know that stress, it triggers a calcium wave, and there are many calcium components here which are involved in the activation of the respiratory burst oxidase. We know that this results in hydrogen peroxide, which is transmitted to the next cell. We know that transporters such as the PIPs, which are involved in transport of hydrogen peroxide into cells, are important. We know that our receptor HPCA1 is important. We also know that this import of hydrogen peroxide opens or is involved in the opening of plasmodesmata, which allows molecules such as hormones to move from one cell to the next. And this signal then is propagated because the next cell receives the signal as a burst of calcium. You have the RBOH and it moves forward. Now, this signaling process is really important in all types of stress, but it tells you nothing about the specificity of the signal. And I realize I'm running out of time, so I'm just going to summarize here. I hope I've convinced you that reactive oxygen species are not just negative factors in cell responses to stress. They're really important in the systemic acquired responses to stress. We know some of the factors involved in the systemic signaling, and we have a lot more to learn. There are phytochrome C. We know the way phytochrome B is involved, which is a surprise. We know the way that signals move from one tissue to another, and we know some of the components. But we know very little really in terms of the overall picture and the specificity to stress. So there is a lot more that we need to learn. And with that, I will finish and just thank the people in my lab, particularly Barbara Karpinska, GII, and Daniel Cartabella, and all my collaborators who have contributed to this work. And with that, I will finish and I will be happy to answer questions.
Thanks, Christine. It's a really wonderful presentation. And maybe I will see a few questions. Where to search? Okay, in chat box. So one of the question, Christine, it is which type of antioxidant used in plant ROS that reduces stress condition?
Right. So one of the things that's always important to know is that if you increase antioxidant levels, and it can be a general increase, it can be antioxidants, ascorbate, glutathione, or any of the enzymes, you reduce the impact of a stress on the viability of the plant. What you also do is limit the signaling. So I would suggest that lots of experiments have shown that antioxidants are important in stress responses. The only thing that I would say to you is that even though we've known for 20 years that if you overexpress antioxidants, plants do better, this has never been commercialized. I have never seen any attempt to increase antioxidants in the field to commercialize it. And I think what happens was, even in laboratory experiments, you see a benefit of increasing antioxidants. When you put them in the field, that disappears. So it is good to increase antioxidants, but it isn't something which could be used, I don't think, to increase stress tolerance commercially.
Thanks, Christine. So for this wonderful response. While the other questions participants are typing, let me ask a few general questions that will not be pertaining to this presentation. So we would like to know, like, what encouraged you to enter this science field, and what are those achievements that you are proud of?
Oh gosh, I think I came into this field by accident. I actually started my PhD working on bananas. I was interested in the ripening of bananas, and we were but if anybody who's listening who's worked with banana fruit will know how difficult it is to work with the fruit. So I did a year of my PhD, and I wasn't getting anywhere. And my supervisor said at that time, look, there's a lot of hydrogen peroxide produced in leaves as a result of photosynthesis. And we don't know what the implications of that, and we don't know how it's removed. So at the very start, I entered the field from complete ignorance, but we were very lucky and able to show that ascorbate and glutathione is important. And it was so exciting, and the implications of the interactions between reactive oxygen species and energy and metabolism was wonderful. I'm proud of lots of things. I'm proud of the start. I'm proud of the way we followed ascorbate, and we looked at ascorbate signaling and glutathione signaling. I'm really proud of the interactions with metabolism, and I'm inspired now by the way that we are looking at the role of reactive oxygen species in stress signaling. To me, that's another exciting avenue. So the interest keeps growing.
Yeah, that's interesting. And also, when I was seeing more information about you in Wikipedia, I was also coming across there is some pathway also that is being mentioned. So, would you like to share about something about that?
Oh, well, I was, I mean, you mean the ascorbate glutathione pathway, which is called sometimes Hallowella asada Foyer. This is it. So, I mean, this is what I did my PhD on. And we showed that ascorbate and glutathione were required for the removal of hydrogen peroxide. And nobody knew that really at that time. And we suggested that these low molecular antioxidants work together to remove hydrogen peroxide. And actually, even though other antioxidants such as thyrodoxins, peroxidoxins have come along, the basic work that we did still holds true. That ascorbate and glutathione are really important, and they function together with other antioxidants. So maybe I think times have changed, but when we did that work, it was possible to have your names associated with a new finding. So that is the pathway, and it was done in my PhD. And I hope that inspires other PhD students that you can do work which is groundbreaking in your PhD, even though at the time we didn't know it. We didn't know it. And we did it in plants. And then the animal people looked at it and they found similar things in animals. So it's a demonstration of something that was done in plants, which was then also found to be true in animals rather than the other way around.
Thank you, Christine. So now when we see Christine as a researcher, we see an accomplished scientist. So we feel that Christine never failed in her experiment. So we would like to know basically whether you failed or not failed. If failed, so how you dealt with that?
Oh, believe me. I think doing science is 90% frustration. Many things don't work. I think you have to be very clever in doing experiments because it's quite easy to follow paths of experiments that actually aren't telling you anything. It's really easy to do experiments which add some information but don't tell you a lot. And many experiments don't work. I mean, the experiments that I showed you with the Pseudomonas and the roots, the first studies that we did were really frustrating because we kept putting the Arabidopsis around the roots and it was strangling, the Pseudomonas around the roots. And the roots were being strangled by the growth bacteria. So we had to learn how to do that experiment correctly in a way that gave us information. Believe me, there are lots of things that we've done that we weren't able to publish because it did, we didn't get the results that we wanted, or the data wasn't definitive. These days how hard it is to publish information. And so yes, believe me, there's a lot of frustration, but yeah, but I am always inspired by the work. So that keeps you going. You have to love it. You have to love what you do.
Thank you. Christine, there are two questions on the presentation. The first question says genes involved in organic acids were upregulated. So was wondering what might be the role of these organic acids.
Yeah, no, that's really, really important. We don't actually know how the changes in transporters and metabolism affects the way that roots process nutrients, and that is I think that's the question, isn't it? Is that what the lady's asking about the roots? So it's a very good question, but I actually don't know the answer. I don't know the benefit of those changes, not yet.
Okay, another question says that why the commercially applied antioxidants disappears from the fields?
Yeah, no, you can't add if you add antioxidants directly to the fields. They're very unstable. You cannot use things like ascorbate in the field. They're rapidly oxidized. And why antioxidants don't work in the field? I presume what happens if you take plants which are modified in antioxidants, the change in the antioxidant is actually quite minor to the other signaling pathways. So I would think that the production of reactive oxygen, I talked about signaling in the apoplast. If you increase reactive antioxidants within the cellular compartments, and you have a lot of hydrogen peroxide produced in the apoplast, perhaps the effect just isn't big enough. So there is too much reactive oxygen produced in the natural environment for the antioxidant to have benefit. I think that must be the answer, but no field trials seem to really work with higher antioxidants.
Thank you, Christine. We know that you have shortage of time. You have to plan your travel ahead. Maybe the last question. Yeah, last question I will ask from my side, that if you want to give some suggestions to the young researchers that may be useful for them to pursue their career in science, we will be welcome those comments, suggestions from you.
Yeah. So I think, you know, pursuing a career in science is, you have to be inspired by the science. If you love the work, then no matter what kind of hardships come along, you will still carry on. It is difficult in life to carry on in a scientific career. You have to be very single-minded. But I think if you are inspired, if you know that what you have done, you've done to your best, the best of your ability, then you will always be proud of what you've done. I think that's really, really important. If you try really hard and you know that you couldn't have done better, then you are satisfied that you've done a good job and you can still carry on. But it is difficult, but it's always look for opportunities, try and find the excitement in science, and try not, I mean, it is, some things are difficult. I still feel very deeply when somebody criticizes my papers or I don't get a grant funded. It still hurts me. It's, you know, but you have to live through it. You have to rise above it and have confidence that, you've done the best that you can and the work is worth doing. That is my advice.
Thanks, Christine, for these words and your responses and sharing your views and some like a presentation. And then most importantly, I really appreciate taking out time. I know that you are busy and then your schedule was very tight, but still you gave us time for this presentation and being our first speaker in this lecture series. So we are very much thankful to you. Secondly, now we know that it is too much cold over there. So now you can have a second cup of coffee there, and then
before I go.
Yeah, close session. And I would like to thank all the audience, also those participants, those were part of. But we know that even like this will be very much used and used by the incoming times also. So thanks, Christine, and thanks all the participants for making it successful. Thank you and have a nice day, and also nice new year also. Greetings, new season's greetings also from our side to everyone. Thank you.
Bye.
Thank you. Manit, please close the session.
Yes.